Blood is a type of fluid connective tissue that carries nutrients and oxygen to various cells in the body and at the same time transports metabolic waste away from the same cells. Vertebrate blood is composed of blood cells suspended in plasma. Blood cells mainly consists of red blood cells or erythrocytes or RBCs, white blood cells or leukocytes or WBCs, and platelet cells. RBCs are the most abundant blood cells. They impart a red color to the blood. In this experiment, we will be covering the procedure and how to prepare a thin blood smear and stain it with an appropriate stain for the morphological and quantitative study of the various cellular elements of blood. For this experiment, we will need the following: blood sample, glass slides, Leishman stain or Wright's blood stain or GM's stain, distilled water, droppers, petri dish or Copeland jar, compound light microscope, immersion oil, cotton and rubbing alcohol, blood lancet or a pricking device. Take 2 glass slides and clean it with 90% alcohol. Disinfect a finger with alcohol and prick with a sterile lancet or a pricking device. Place a tiny droplet of the blood at the center of one corner of one of the glass slides. Using a second pre-cleaned glass slide, touch the drop of blood while inclining the slide at a 45-degree angle. Now gently and briskly move the inclined slide towards the other end of the slide containing the blood drop in order to obtain a smear. A properly smeared blood appears roughly tongue-shaped as seen here. Allow the smear to air dry for a minute or so. Place the slide in a petri dish with the blood smear facing up. Add several drops of the Leishman stain to the blood smear and cover the petri dish. Alternatively, you may fill a Copeland jar with Leishman stain and immerse the slide in the stain. Stain for about 1 minute and then add twice the volume of distilled water to the stain. Allow the water and the stain to properly mix and keep it aside for about 10 minutes. Drain the slide and wash with distilled water. Proper staining results in a blood smear that is rose pink in color. Air dry the slide while keeping it in an inclined position. The slide is now ready for observation under the microscope. Observe and study the various blood cells under 40x and 100x objective. For 100x, place a drop of immersion oil on the slide. Gently lower the 100x objective until it touches the oil surface. Hereon, use the fine adjustment knob for focusing. The following microphotographs show the various cellular components of human blood.